Okay. Welcome back to Longevity 0 to 1. In the next 90 minutes, we will talk about aging biology. This is roughly an agenda. We talk about what is aging and why we age. And then we will go through a couple theories of aging. And then we'll talk a little bit about how aging varies between species and between humans. And we end with immuneaging. So we can welcome Sheekey back to the stage.
Hello, so I'm back again for a couple more sessions. This time, even bigger questions. What is aging and why we age? So firstly, I thought I would start with what is aging, and unfortunately, there's no way I can really talk about this without probably offending, well, not necessarily offending, but causing some disagreements amongst the field. As if you ask a group of researchers in the aging field what is aging, it's very likely that you would get very different answers depending on who you speak to. So that's just a bit of a preface. But I actually thought I'd start a different way, which is at least we can describe certain features about aging. And given that yesterday it was the king's coronation, I thought I'd show some pictures of our king. And I think we can all quite easily describe certain features that happen with age. Our likelihood for different diseases increases. There are certain physical things that we notice. We become stiffer, we get wrinkles, hair graying. But in terms of understanding ways to intervene in aging and eventually cure aging, we kind of have to go beyond this sort of very descriptive features and have a better mechanistic understanding about what is aging. And so even my briefish career in aging research so far, my thoughts on what is aging has also changed over time. But one thing that I generally tend to say if someone says, what's aging, is that it's kind of the progressive deterioration of an organism over time. And so we see things such as loss of muscle mass, loss of strength, memory loss, decreased fitness and function, and all of this increases our likelihood of death. And so ultimately you can somewhat summarize that in that aging is something that results in the accumulation of damage within the system that enables us to live. And so that was my very brief intro into what is aging. In fact, what's probably a more interesting question, which we'll have more discussion on throughout the rest of today, is why do we age? What is actually underpinning this process? Is it something stochastic, or is it something that's been programmed? And how can we use that information to then develop different ways to intervene? So if you've done any research about longevity or aging research, it's very likely that you've come across the so-called hallmarks of aging, which there's been some backlash as to whether this is the best way in which we should be thinking about aging, but I still think that it is a good way to sort of ease into the question about why we age, partly because it sort of covers a lot of different topics that we'll discuss probably in the rest of these few days. So this is actually an out-of-date slide that I took from a previous presentation, which says that there are 9 different hallmarks of aging. And I'll take a look at some of them in a second, but actually it's been updated recently to include a few more features. And so I'll actually just go to the up-to-date version. In fact, there now seems to be 12 different hallmarks that help to describe the different things that we observe with age. And so these go from so-called primary causes of aging, which include things like the accumulation of damage in different molecules, so things like DNA damage, mutations in DNA, protein damage, where proteins become dysfunctional or the turnover of proteins becomes dysfunctional, as well as so-called antagonistic hallmarks. And so these include things such as cellular senescence, so when a cell enters into a state where it stops doing what it usually does and can become inflammatory. And so cellular senescence is something that I actually study myself, so I'm not going to go into any depth now, but if you want to know the ins and outs of cellular senescence, then I'm happy to do so at a later time. This also includes mitochondrial dysfunction. We spoke about earlier how mitochondria is important for generating ATP, the energy source of a cell. Mitochondria do much more than that. They're also linked with how a cell regulates cell death, and a whole series of things such as heme biogenesis. So That's mitochondrial dysfunction. And then there's also so-called integrative hallmarks, which includes things such as stem cell exhaustion. So stem cells being cells that have the ability to keep replicating, but also the potential to differentiate and turn into certain cell types. So this could be like a skin cell, a muscle cell. And so this appears to be both loss of this ability of stem cells to proliferate, but also just the loss of stem cells in general as we age. And there's updates that came out this year about these different hallmarks. They've also now included things such as our microbiome. And so our microbiome is found in different places within our body. Most commonly, people think about our gut microbiome. And I was actually discussing with people earlier about this. There's a lot of things we don't know about it, but they produce a lot of metabolites as well, which seems to have massive influence over various different systems within the body. Essentially, what I'm trying to say is that this is just a very descriptive thing about different features that we can see with age, but that doesn't still really get to the point about why do we age. So this diagram just is a better view about how we can split it down into these different hallmarks, but actually there's massive overlap between them. We're not sure what the initial trigger is, we're not sure how they're interconnected, and we're not essentially sure where is the best target to intervene. So because I have time to do so, I thought I'd just go into a bit more detail about some of the observations that have been made into these different hallmarks to actually show some evidence for it. But I know as well that there'll probably be some overlap with some of the talks that you'll have later today. But this that I'm currently showing goes into some detail about a recent publication that came out last year that essentially took cells from different species and looked at their mutation rates and correlated it with the maximal lifespan of that species. And so what you can see firstly is that even, also as we know, different species live for different time periods. And the question is, what does that tell us about aging if there's this massive spectrum of lifespans seen across different species? Why is it that we live to about 75 years while mice have a lifespan about 2 to 3 years? What can we learn about those differences? And what this study effectively found was that there's a very nice correlation between the mutation rates with maximal lifespan in species. So, what if this works? Slowly. So things like mice, they have a much shorter lifespan than we do, and it's as suggested from this paper is possibly because they have a much higher mutation rate. But this is just DNA damage. Similar sort of correlations can be found by looking at lots of different features of aging. And so it kind of gives us an insight into what aging is, and it's also why different aging biologists have different viewpoints about what are the major causes of aging. But I still think the big question about why we age is unclear. And that comes back to this question that I mentioned very briefly, which is aging just purely a random stochastic process where we accumulate damage? Or is it programmed? Is it some reflection of the fact that we're built to develop and to reproduce, and that as a consequence, there's these side effects such that there's a de-repression of certain pathways that are responding to damage? I realized that sentence didn't make much sense, but I'm trying to keep it. I can explain in more detail afterwards if you want to know. I also think I'll be talking a bit more about it tomorrow. But essentially, in terms of asking the question, is aging programmed, there was a growing group or consensus that actually maybe aging is a sort of developmental, a reflection of there being flaws within the developmental process, and that evolution basically has been adapted such that we can grow and then reproduce, and that whatever happens after that period is not going to be selected for. And therefore, can we understand what this developmental process is that's running and find ways to slow it down and therefore potentially prolong or postpone the period in which we age? And another somewhat support for that is that we can look at so-called DNA methylation marks and develop these methylation clocks that you may have heard of that seem to correlate with age. And so methylation in particular happens on these cytosine residues of DNA, and there are now these epigenetic aging kits that are meant to give you a biological age, which has been shown to correlate with chronological age. and give an indication about the rate of aging. So what this figure is essentially showing is that someone who is aging faster has a methylation age that's greater than their chronological age than would be expected, and vice versa. In the blue dots here, someone who's living longer, potentially due to lifestyle or genetic differences, has a slower tick of this methylation clock. And so it goes at the moment, we still don't know if these methylation marks are causal to aging or they're just correlating with aging. And that raises the question as, if there is this such nice correlation, what does that tell us about the aging process? Is it something that's predictive and that we can therefore use that to infer where to intervene, or is it just a stochastic process? Another way that you can think about aging, someone mentioned earlier, or after the Q&A, the last session was about using metaphors, and so this is another metaphor, I'm sorry. But I quite like it, is that we can think about aging as being akin to gravity. It seems to be something that is inevitable at the moment. So far, we haven't found a way to cure aging. But there are potential different ways in which we could intervene in the aging process. One are so-called geroprotective therapies, so being able to slow down the process. Alternatively, can we find rejuvenative processes where we actually refast the aging phenotype? But ultimately, there's still this continual pull, which is the aging process. We might be able to rejuvenate, but it would still age. And that's whether we can rejuvenate faster than we age, we might actually be able to cure aging. But yeah, I just think this is a nice way to sort of visualize some of the different ways in which we try to understand aging. But it's actually quite an important question, and it's quite challenging to explain what is aging when there is no general consensus. But it's something we really should try and define, because once we have a definition and agreement about what aging is, it enables us to better ask experimental questions to then work out why we're aging and how best to intervene. So the other thing that's worth mentioning briefly, I know that there'll be some talks later, is about genetics and intervening in aging. There's been a whole series of studies now that suggest that intervening in aging is possible, yet many people ask, well, if we know this to be true, then why have we still not seen actual human therapies for aging? And so a lot of work has now understood that there are genetic differences, like centenarians within human populations, where there are certain genetic variants that seem to give people better disease resistance, and how can we interpret that? But also just from classic model organisms, we can also find genetic mutations that seem to extend lifespan. So this is now a somewhat famous study by Cynthia Kenyon, where she identified a mutant in worms where it extended their lifespan. And from that, we then sort of uncovered the function of what this gene was, the protein it was encoding, and that linked to nutrient sensing, and this led to us a better understanding about metabolism and food intake, and also certain supplements such as rapamycin. And so we can use genetics to understand essentially what different proteins are doing and how they're playing a role in the aging process. But I still don't think we fully understand it, essentially. This is just to show that there are even, obviously we're all genetically different, and we can use that information to understand why some people age at different rates. So this heat map, if you can see it, is basically showing genetic variants from different individuals and correlating that with their parental lifespan, whereby you can see a negative correlation between having a longer lifespan and the risk with different diseases, while there's a positive correlation between the different diseases. Long story short, there seems to be genetic variants that, if we can understand the function of those variants or their impact, that could inform us more about the aging process. And then that also leads on to lifespan. A lot of people like to come up to me and ask, what do I do for longevity? And I mean, there is some exciting research being done. I'm just going to sort of throw out a couple of different things, in particular about food intake, food quality, food timing. And so far, like from model organisms, we can see that calorie restriction, so reducing food intake, seems to be pretty robust across different model organisms in terms of increasing lifespan. But then the question is, how could we then translate that to human therapies or find ways to mimic it? And in terms of mimicking, We can come back to things such as rapamycin, which inhibits a protein that's involved in regulating cell growth within a cell. And so essentially there seems to be so much exciting research in both genetics and lifestyle in terms of all the different hallmarks of aging, yet we still don't really have an agreement over what aging is or why we age, which I want to say this in an optimistic way, that I feel like we are making progress without sounding too pessimistic. But it's definitely, hopefully sets the scene for a lot of the discussions we'll have throughout the next few days. Yeah, hopefully that's been somewhat useful. And this is just a really abstract diagram I thought I'd throw in to show that, as well as considering just our single cells. I spoke earlier about the different levels in which we're built, from cells to tissues to organ systems, but we're also in constant communication with our environment. And actually, there's many different levels that maybe we should be considering to understand this aging process. So yeah, hopefully that's given you some sort of gentle ease into aging research, and hopefully the next few talks will give you more in-depth detail about different theories about why we age, or what is aging.
All right, everybody. Yeah, I just got here late last night, so I'm just getting used to the Zuzalu vibe, and I must say I'm rather enjoying it. I always find that it's easier to give talks to geeks, which seem to be widely represented here. And so I'm hoping to be able to give you some good information. I believe I'm also giving a talk the day after tomorrow. Yeah, so I'm going to go into more depth about the work that I've been doing over the past 20 years or more in that one. So this is going to be a little bit more of a top-level one. And I've been asked to talk about information theory and damage accumulation theory, and I will have some things to say about those choices of words. So first of all, yeah, well, so this is basically what I'm going to say about the information theory. First of all, that the concept as it's currently discussed by the chattering classes really arises from a book that was written by my friend David Sinclair about 5 years ago, in which he introduced the concept. And that's what I'm calling the new meaning. But if you took anyone in the field any time before that, and you said, what does the phrase, the information theory of aging, mean to you? They would have said something completely different. So I thought I'd just go through both of them. But first of all, as some of you know, I tend to get a little angry sometimes, so I thought I'll do that. There are basically three things that I don't like about the phrase, the information theory of aging. They are information, theory, and aging. And I shall talk about those. So first of all, aging. Well, we've just heard, right? I mean, as Eleanor quite correctly said, you know, if you ask 10 experts what aging is, you'll get 11 different definitions. It's absolutely fucked up. I mean, we've been worrying about aging since the dawn of civilization. You would have kind of thought that we would have figured out how to define it, but no. So something's got to be done about that. And in particular, the relationship between aging and disease. You know, when it comes to revolution, when I rule the world, the words aging and disease will not be allowed to be used in the same sentence. Because if you say that aging is a disease, it has an enormous amount of collateral damage that you do, that people assume by what you mean by it. And if you say aging is not a disease, it's even worse. So, you know, that's just not allowed. Today it's become fashionable among the experts in this community to say this thing here, that aging is a risk factor for chronic disease. Now, exactly what might that even mean, right? First of all, aging is a process, right? How can a process be a risk factor? Obviously, what they actually mean is age is a risk factor for chronic disease, but that won't do either, because if you say that, then of course you've got to say, what do you mean by age? Do you mean actually how long ago one was born, or do you mean what we like to call biological age? In other words, basically, how much damage do you have in your body? Eleanor was very correctly defined aging herself as like the accumulation of damage. So really we mean biological age. But then biological age simply is the accumulation of the early stages of what becomes chronic diseases. So all that this statement really means is that being nearly sick is a risk factor for being actually sick. Well, I mean, no shit, Sherlock. I mean, really, that's not exactly clever, right? So, you know, it annoys me, as you can tell, that people who are not trained in like, you know, logic and maths and stuff say things that are so kind of unfalsifiable. Then the word theory. The word theory is even worse, because the fact is, theories are things that are either true or false. You know, you have hypotheses and theories when you want to understand nature, right? And we're not about understanding nature, we're about manipulating nature, we're about doing something about aging. So having theories of aging kind of doesn't really, it kind of misses the point. And there are very, very few people, even now, there are very few people in the field who actually think like technologists, like engineers, and they actually want to try and do something about aging as opposed to simply understanding it. And then, yeah, I'm not terribly happy about the word information either because, you know, what we really care about is the manner in which information is embodied. And there are lots of ways in which information is embodied in a living system. And that brings me on to the old meaning of the information, quote, theory of aging, which is all about entropy and the second law of thermodynamics. People pointed out some time ago that the body is a machine. Well, duh, yeah. And with moving parts, which means that, you know, it's a fact of physics that it's going to damage itself. It's going to accumulate damage as a consequence, an inevitable consequence of its normal operation. And so people said, oh, well, in that case, that must mean that, you know, it's all downhill and there's nothing that medicine's ever going to be able to do about it. But of course, that's a misuse of the second law of thermodynamics, which is all about closed systems, systems that do not export entropy. Open systems do. And, you know, the fact that babies are born young is, of course, a perfectly good example of this. This is because one can use energy to partition entropy so that there's not much entropy in a baby. And that's all you need to do to explain why aging is not something that's off limits to medicine on account of physics. In fact, really, if one wants to talk in those kinds of forms of words, one can perfectly say that medicine, certainly medicine against the health problems of late life, is all about augmenting the machinery that we naturally have to export entropy, without which we would definitely not be alive at all. It's absolutely essential in a living system to be able to export entropy pretty comprehensively. And the reason we age is because that export is not 100% comprehensive, and medicine is about filling in the gaps. So the new meaning. So I mentioned David Sinclair wrote this book, and he's written a few papers and done a few experiments that have given some weight to the idea that a particular mechanism for the accumulation of damage plays a role in all this. He pointed out, quite correctly, that we can use the analogy, which came up in the session earlier on, that the genome is basically a computer program. It's software. It's even digital. That's fine. In fact, probably the best way to describe it is as our operating system. So then, what is the epigenome? So the epigenome is these decorations on our genome, these changes to the methylation state of certain bases, and also other things like wrapping the DNA around certain proteins and such like, which determines which genes are turned on and turned into proteins in a particular cell and which ones are not. So from a computer science analogy, probably the best way to say this is the epigenome is the data area of that program that is written and read and causes the program to proceed. And David puts forward this rather interesting idea, which makes perfect sense, that there could be a contribution in this of the degradation of the epigenome, in other words, corruption of the data, arising from a system for repair, a system of repairing the actual software. You see, the software, the actual sequence of chromosomes, undergoes damage all the time, and some of that damage is quite difficult to repair accurately, and the manner in which it is repaired is somewhat error-prone and makes mistakes in the sequence occasionally. But what David pointed out was that in addition to that, there will be changes to the decorations that I just talked about, the epigenome, and that this may accumulate over time. Now, this assumes that we don't have systems to repair the epigenome. And that's pretty dangerous, because another thing that's very popular right now, and indeed also David works on this, is something called reprogramming, epigenetic reprogramming. This is a phenomenon that was discovered as a technique in biology only about 15 years ago, and the person who discovered it, Shinya Yamanaka, already got the Nobel Prize for it. Essentially, it recapitulates what happens in the fertilized egg, and very shortly afterwards, when you've got two very specialized genomes, the genome of the sperm and the genome of the oocyte, which come together into a cell and have to kind of be wiped clean into a state that can, obviously in unison, develop through the early and then subsequent stages of development. And basically the tricks that Yamanaka discovered recapitulate that. Now, what therapeutic use might that be? Well, there are various uses for creating different types of stem cells, and I'm not going to go there right now. But in addition to that, there is the possibility that we might be able to just nudge things using this technique in an adult organism. And that's what's now called partial reprogramming. You'll come across partial reprogramming a lot in the general audience coverage of aging research these days, because it's become certainly the best funded of the areas within aging research. The biggest company in this space, Altos Labs, is mostly focusing on it. So are a couple of other companies and big groups. So it's a big thing. So the idea here is that you throw in these factors that could potentially turn your body into protoplasm, which is probably not what you want. I don't know about you, but I'm quite fond of my central nervous system, and I don't want it like de-differentiated. But the point is, you could do it just a little bit. And what you might achieve with that might be to nudge some cells that should be stem cells, but they've become a little bit too, not quite stem-like enough, and nudge them back into being as stem-like as they ought to be, so that they'll be more regenerative, so that generally, you can rejuvenate the body in not every way, but in a lot of ways. And this is a clever idea. However, really, it's dangerous, because what it requires is for cells to still know what kind of cell they're supposed to be, essentially to repair the epigenetic slate cleaning that you didn't want to do, while at the same time, of course, not restoring the randomness, whether by the mechanism that David described or by other mechanisms, that has built up over time. And, promising results have been found, both in cell culture and in mice. I'm personally still very much, not exhaling when it comes to this, because there are good reasons to believe that the impact on a long-lived organism could be quite double-edged. In particular, that long-lived organisms are full of cells that have accumulated spontaneous mutations that are heading them towards cancer, and that may actually be exacerbated by this technique. But that's just a concern. It's not something that we actually know. So that's all I'm really going to say about information theory. It's a big topic right now, but whether it really means anything is it's still open to debate. So I'm going to do a little bit more of a rant now about the word theory. The concept of theories of aging is not actually very much talked about in the field these days. It's kind of gone out of fashion, and I was actually a little surprised that David revived it, because people have come to realize that it's just like you need to define what you mean by a theory of aging. So really, you can talk about strong theories and weak theories. A strong theory of aging is basically the hypothesis that there is one particular mechanism of damage accumulation and eventual pathology that arises from it that is the most important, that kind of is dominant in the decline of function and eventual death of organisms. And so that's one definition. A weak theory simply says that such and such a mechanism is a contributor, not necessarily the dominant contributor. And way back in the 1980s, a pioneer of calorie restriction research named Roy Walford, who did all his important work before almost all of you were born, he said the problem with calorie restriction is that it supports all theories of aging. And of course what he meant was all weak theories, because it's true. Basically calorie restriction slows down pretty much everything, and that's why it results in life extension, at least in short-lived species. But neither calorie restriction nor anything else supports any strong theory of aging. So since that's kind of the conclusion, we see that basically aging consists of the accumulation of a wide variety of different aspects, different types of damage, and there's a certain amount of crosstalk between the mechanisms of accumulation of different types of damage, but only a certain amount, then, you're going to have a lot of weak theories that are true, and you're going to have no strong theories that are true, and so kind of that's that. So the whole concept, the whole terminology of theories of aging has kind of gone out of fashion now. But the other thing I wanted to talk about before I go on, and because I'm talking to technologists here by and large, is something that really didn't occur to me until my early 30s, when I started really looking at aging and understanding why the people who were working on it were having such trouble getting their hands around it, is that there is a vast difference between the ways of working and thinking that work well in basic science versus technology. So what I mean basic science, I mean understanding nature, trying to figure out how nature works, versus manipulating nature, which is what technologists do, trying to make nature work differently. The thing about understanding nature is almost everybody who does it is what they like to call curiosity-driven. In other words, they find things out for the sake of finding things out. And a large part of the way I got into this field was when I met and married a biologist at the age of 20-something, and I was a perfectly self-respecting computer scientist until then. But then I obviously we were talking about biology, and she was a lot older than me. She was already a full professor at UC San Diego. I would say things like, you're not interested in aging, are you? And she would say no, and I would say, why not? And she would say, well, it's just decay, isn't it? What fundamental truths about the universe are you going to understand by studying decay? And I would say, well, sure, but it's bad for you. And she would say, well, that's not my problem. And I would say, well, it kind of is. And that would be that. It would be what we would get. So that's actually a large part of why I decided to switch fields. But the thing I wanted to highlight here, which is important for any of you who might be thinking of getting into this, is that the way you think about what experiment to do next, what thing to try next, is completely different if you're trying to understand nature versus manipulate nature. If you're trying to understand nature, you really don't want to take big leaps of faith. You want to find the next really, single small step in terms of distinguishing between competing hypotheses. And you want to have as few confounding factors as possible. You want to have experiments that are really likely not to mislead you by their results. Whereas if you're doing pioneering technology in anything, whether it's medicine or anything else, then you don't do that. I mean, basically what you do is you come up with a concept where you're putting a bunch of established things together in a new way, and you have no idea whether it's going to work. It's really just a big leap of faith. And that's how technology works, and I'm very much a technologist, and the real reason why I was able to make a contribution when I started in this field 20-odd years ago was because I was more or less the only technologist. So this is basically what I said back then. I said, right, well, aging is the accumulation of damage. I'm describing it here in 3 words. That metabolism, in other words, the network of processes that the body undertakes to keep us alive from one day to the next, generates these changes to the molecular and cellular structure of the body and its composition. And those changes are initially harmless because the body is set up to tolerate a certain amount of those changes, but it's only initially harmless. Eventually, the amount of damage exceeds the amount that the body is set up to tolerate, and that's when we get the health problems of late life, pathologies of late life, both physical and mental. So the goal here, as technologists, our goal is to separate metabolism from pathology, right? It's to allow us to live in a healthy, useful state for longer, even though we are still performing metabolism. In other words, we're not in hibernation. And of course, originally, in the distant past, the understanding of the difference between the health problems of late life and other health problems like infections was not understood at all. And so geriatricians tried to treat these things in the same way, to just kind of eliminate them from the body. And of course that works quite well with infections, and it doesn't work nearly so well with things that are the product of normal metabolism. So that was a failure. And it took until about a century ago before anyone really got the message. And then about a century ago, early 20th century, a few people started to realize this is just not going to work. This is a mug's game. So they realized we needed to be more preventative. And that's where the whole field of gerontology arose. Because people looked at nature, again, coming back to one or two of Eleanor's slides, they said, well, look, we've got this enormous variation in the living world in the rate of aging. So maybe we can try and study that and figure out what's the basis for that variation. And once we understand it well, maybe we'll be able to translate that into some kind of intervention that makes metabolism run more cleanly, and in other words, generate damage more slowly, and bang, we'll be able to live longer without getting sick. And that has also been a completely spectacular failure. There have been some false dawns, in particular the paper that Cynthia Kenyon brought out in 1993, which Eleanor mentioned, was the source of an enormous focus by a large number of scientists on genetic and pharmacological manipulations that would, in one or another way, mimic the phenomenon of life extension from calorie restriction, which it turns out doesn't work nearly so well in long-lived species, and there are good reasons why that's the case. I'm going to tell a little story about that paper, actually, because it'll give you a feel for how hard it is for biologists to understand aging. So the authors on that paper are Cynthia Kenyon and 4 others, and in marked contrast to the normal situation with papers in biology, Cynthia, the senior author, is listed first rather than last. The reason for that is because all the other authors were rotation students. That means first-year PhD students in the US who haven't even chosen a lab that they're going to do the actual PhD in. And the reason that was the case was because none of the actual students or postdocs in Cynthia's lab were willing to participate in the experiment because they thought it was crazy. Because the dogma was that you couldn't possibly do this. You couldn't possibly extend life by one gene. So, this is a measure of how paradigm shifting that was. But as I say, it turns out to have been a bit of a false dawn, at least from my perspective, because you can't extend life very much by that sort of mechanism in long-lived species. Other than that, though, people have tried a whole bunch of other things, of course. We've basically got nowhere. There's just too much linkage. The creation of damage is just too intrinsically intertwined with the processes that metabolism needs to do to keep us alive and healthy from one day to the next. So the maintenance idea, this third idea, is really what I'm best known for in the field, because I was the first person to come along and say, well, hang on, there's a third way to do this. If the goal here is to separate metabolism from pathology, as I said, and we figured out that we can't do that by separating damage from pathology, and we also can't do it by separating metabolism from damage, then let's look at this other option, which is to periodically repair some of the damage so that you end up having damage created at the natural rate, but still not reaching the threshold that causes it to be bad for you. And I put some flesh on those bones in a paper that came out 11 years before hallmarks became cool. So this is the original hallmarks paper, I guess. And honestly, I still prefer this classification to all of the upstart ones, largely because of what's on the right-hand side of this table. So on the left-hand side are things that are very reminiscent of the hallmarks that we see today. But on the right-hand side, there are corresponding methods for going about fixing these problems, actually doing the damage repair. And I feel that the more recent attempts to classify the processes within aging kind of miss the point, because they don't have that. They only have, they're a classification of the things that happen without really giving us any kind of leg up towards how to think about fixing them. So this is the kind of classification that I think we should be more focusing on. And what it leads to is a concept that has not only led to the name of my new organization, LEV Foundation, but it's also a lot of the reason why a lot of the people in this field have got into it, especially people who have their training in maths and computer science and so on. It's the concept of longevity escape velocity. If you develop a therapy, which I've already explained we won't, but even if you could, that slows aging down, then what you're doing is you're slowing down the creation of new damage that has not yet been created, but you're not getting rid of the damage that's already being created and that your body is carrying around. So if someone's already, let's say, 60, and they start taking a therapy that slows down, that halves the rate at which they accumulate new damage, then, well, it's going to take a bit. It's going to take maybe, 20 years rather than 10 years for them to get to biologically 70 and thus start to get sick. But they're still going to get sick quite soon, which is a bit of a shame, really. Whereas if you're doing damage repair, then you don't really have that problem because you can buy yourself time. Supposing we have a therapy that repairs even a third of the damage in a 60-year-old and takes them back to biologically 40, shall we say, then you've bought the scientists 20 years to figure out how to do it better the next time around to the same people. So you end up in a situation where the therapies have to be improving over time, but only at a finite and indeed a diminishing rate. And the people who are getting the best therapies at any given moment will actually never get enough damage in their bodies to make them sick, however long ago they were born. So if you think about that in the abstract, then it does have quite a lot of similarities mathematically to the concept of gravitational escape velocity, and that's where the name came from. This thing at the bottom that I've written is perhaps a slightly overly complicated statement, but I'm sure you can get the concept that essentially once we get those first couple of decades of increased life, in other words, postponed health problems of late life, so that we get people aged, in their 140s or whatever, just occasionally, same as we have right now, occasional people getting to 115 or whatever, then we've done the hard part. That by that time we'll be buying time rapidly enough that there's no reason why people shouldn't live indefinitely. The number 1,000 is just there for rhetorical purposes, really, but it's actually an extraordinarily conservative number, if you think about it. If you're living in the Western world these days and you're at, let's say, you've just reached the age of 26, your chances of not reaching 27 are less than 1 in 1,000. So if that number stays the same as you get progressively older, then of course you're going to have a 4-digit lifespan on average. And that's not accounting for the fact that, of course, causes of death that do not have to do with how long ago you were born will also be going down very rapidly. So I just thought I'd spend the last 5 to 10 minutes talking about the work that we're doing at LEV Foundation. And this is the main project. I'm not going to talk about all the things we're doing, but our flagship project is something that really embodies the race to getting to longevity escape velocity. RMR stands for Robust Mouse Rejuvenation, which is kind of the equivalent in mice. And what we're doing, it will not surprise you from what I've already said, is we are combining rejuvenation therapies. We're taking things that address particular types of damage and have already been shown in the published literature by other groups to have some, at least modest, efficacy in postponing health and extending life, postponing health decline and extending life. And we want to ask the question, what happens if you stick them all together? And of course, we've got lots of controls. We're like looking at 3 out of 4 of them and so on. And the point is we're starting late when the mice are already in middle age. The kind of mouse that you use for this experiment tends to live on average to about 2.5. So we're starting when they are about 1.5 years old. We want to double that. We want to take them out so that they live on average 3.5 years, if not more. And of course, we're measuring lots of aspects of health as well. Really, this is the final stage of demonstrating that aging is absolutely something that can be brought under medical control. The further we can push out mouse health span and lifespan, the more impossible it will be for the naysayers to come along and say, oh no, this is never going to work in humans. I won't go through this in too much detail, but basically we're doing these very diverse treatments. We're doing it in a very sophisticated and careful way so that we can get as much information as possible from this one experiment. Things like dissecting whether the treatment is beneficial, whether the way the treatment is being delivered is detrimental, things like that. So I'll be here for the rest of this week and actually for the next 10 days, and so will my main scientist, Caitlin Lewis, is there. Put your hand up, please, Caitlin, who has designed this with me and is absolutely indispensable to me. So yeah, we can talk to either of us if you want to know more details about this experiment. I'm just going to talk a little bit here about what we are checking for in terms of health. It's really important to understand that lifespan is not what we work on. We work on health span. We work on postponing the health problems of late life, but we're very happy that increasing health span has the side effect of increasing lifespan. And that is something, for those of you who, because this is supposed to be at least largely an advocacy session, right? That's something that I really want to impress on all of you, that people say terrible, terrible things about the idea that increasing lifespan is a bad thing. You know, and people who work in the field will often try to apologize for this side effect and say, oh no, we just want to increase health span, you know. It's appalling, it's absolutely outrageous, but you have to be ready for that if you're talking to people who are somehow opposed to this kind of work. Actually, while I'm talking about advocacy, let me say something parenthetical here. So I complained a bit about the use of the word aging earlier on. I have an advocacy hack for all of you. So, if you get into this and you're interested in helping to change the world by changing people's opinions, then from time to time, or in my case every fucking day of my life, you will have to have conversations with people who will try to make excuses for aging and say that it's a blessing in disguise, or say that it's somehow woven into the fabric of the universe and the idea of doing anything with medicine is nonsense. And they will try to tell you why they think that. And in general, you will immediately be able to see that what they're saying is complete nonsense. But then you've got to convince them that it's complete nonsense. And that turns out to be quite tricky. You know, one's hit rate tends to be rather low. So here's my hack. My hack is, when someone says something to you that is obviously complete bollocks about aging, ask them to repeat themselves, but without using the word aging. This is the way to get around this problem that Eleanor first pointed out, that everybody has different definitions of aging. People just don't have one definition of aging, especially people who think aging is a good thing. They will have mutually contradictory definitions of aging in their head. So this is a really neat trick to help you to understand how to pick people's arguments apart. Get them to admit what their argument really is by not using the word aging in what they're saying. It works well. All right, back to the experiment. So we're doing loads of things. So basically this little diagram over on the left, it more or less says, we are looking at whether the mice look old, like whether they're losing hair or whether their spines are getting curved, whether they basically become hunchbacks, which happens to mice quite uniformly. We also look at how they behave, things like, we do memory tests. We take blood draws and we ask, what's their glucose tolerance like, things like that. And we also actually kill some of the mice. They don't count in the lifespan experiment, you understand. But we kill some of the mice at certain points so as to determine things that you can't determine unless you kill them. We look at their organs really closely. So lots of stuff we're doing, and we kill them at various time points. Actually, this is an innovation that I brought in, which I'm quite happy with because it does seem to be not to have been done before, but people seem to be quite positive about it. The idea is here that most experiments you will choose to kill your mice for those experiments, or indeed to do the other tests, at particular time points determined by chronological age, how long ago the mice were born. And we're doing it on the basis of what proportion of the mice have died already, because this kind of has a better signal-to-noise ratio. You're going to factor out things that are already correlated with each other. You're not going to be measuring them so well. So that's quite important. All right, so I think I'll stop there. This is the name of the foundation, as I mentioned earlier. We've obviously got a website. There's plenty more information on it about that experiment I've been talking about and about all the other experiments I'm doing. We also have a conference coming up in August the 17th to 20th. I should have bloody put that slide up with a QR code and everything. But anyway, yes, it's in Ireland, in Dublin, which is a great party town. Very much like the people who organise this thing, I believe in emphasising the recreational component of conferences. It's where the work gets done. And Dublin is a fantastic place to have such a thing. So hope you can come there. But as I say, both Caitlin and myself will be here for the next 10 days, so plenty of time for you to buttonhole us and buy us drinks and ask us questions. Thanks very much.
Discuss intra and interspecies. Much closer. Is it better? Okay. Intra and interspecies variations in aging trajectories and lifespans and potential determinants of these variations. Better? 1, 2, 3. So perhaps the most widely recognized hallmark of aging is increased mortality rates. I don't mean the accumulative risk of mortality that goes up by definition, but rather the mortality risk at any given moment. And some would go as far as saying that this is the defining feature of aging, and that unless that applies to you as an individual or a species, unless that applies, you are not in fact aging. And until recently, there hasn't been very good measures of aging besides analyzing these mortality rates. So demographic aging has been a prime metric by which you would compare populations and species with regards to aging. And humans and most familiar species age in a nonlinear way. And this is described by the Gompertz law, which means exponential rise in mortality rate. Can you see the pointer? Yeah, so this is the Gompertz law. It's the chance of death per unit time. And this G, the decay factor, it is such that mortality doubling time is about 8 years, which means that every 8 years you become twice as likely to die. So on your 38th birthday, you'll be twice as likely to die compared to your 30th birthday, and so on until you actually die. And actually, if it weren't for that, if mortality rate remains as in our 20s, life expectancy would be around 1,200 years, not my calculation. And Gompertz law is not a law per se. It's not a law of nature. It's not universal, though many species show this pattern. This is a Nature paper from 2013, which described mortality curves for an array of species. And you can see in the shaded, actually doesn't show that well, the shaded gray thing is the survivorship, which depicts the proportion of individuals that are still alive at any given time. And maybe more important, the red curve, this is the mortality rate, starting from around puberty. And you can see on the top left corner, these are humans from Sweden and Japan. You see that it's exponentially growing. And the light brown ones are mammals, and they follow a similar pattern to humans. But if you look further down, there are some non-mammalian species where this red curve is almost flat, even decreasing. So you know trees are in shaded gray. So trees, they never stop growing, and they become more and more robust and actually become less prone to dying. So they sort of experience the reverse of the familiar form of aging. And overall, you can see that there's highly diverse patterns of mortality. And another thing to notice is that the blue curve that depicts fertility, when this goes down, the mortality shoots up. And this observation is, it reflects the idea that natural selection optimized organisms to live only as long as they reproduced and not much longer. And it's particularly interesting to look at these flat curves, and especially in vertebrate species, which is a group that includes humans. This is called negligible senescence. And one paper from last year found that by surveying data from zoos and aquariums for turtle and tortoise species, they found that most of them actually exhibit negligible senescence. And a paper published back-to-back showed similar patterns in wild populations of reptiles. And yet another one, a paper from the alphabet company Calico, showed that naked mole rats, which are a species of subterranean rodent, also exhibit negligible senescence, and this is especially interesting because these are mammals. But okay, there's also a limitation I want to mention. So these studies naturally have very few data points for very advanced ages, because even if the organism doesn't age, you don't have a lot of individuals that actually reach very old ages, so the data becomes kind of scarce. So it's possible that at some point mortality does begin to climb. And actually I would expect that for naked mole rats, for instance, because they do show some signs of aging, they become more wrinkly. So that might manifest as like overall frailty at some point. But at least according to the data they have, they are not aging. Maybe a true immortality also exists in multicellular organisms. This is the hydra, and it's well recognized to be non-aging. It is a freshwater dwelling invertebrate about 1 centimeter long. It's named after the many-headed Greek mythology monster, the hydra, because it can regenerate tentacles, and it has a completely flat mortality curve. And another non-aging organism is the so-called immortal jellyfish, which has a unique feature that under specific stressors like starvation, it can revert back from the medusa phase of its life into the polyp phase. So it can actually iterate over its life cycle and thus evade aging altogether. Maximal lifespan is another metric that varies greatly between species. It derives from your basal aging rate, the basal mortality rate plus aging rate. So it's highly related to aging. And in mammals, it varies from about 200 years for bowhead whales. That's based on a harpoon found embedded in one specimen, a very old harpoon, and down to about 1.5 years in some species of shrews. And for fish, there's an even wider range. And recently, based on carbon dating of eye lenses, a specimen of Greenland shark was measured to be around 400 years old, which makes it the longest lived vertebrate known. This was on the cover of Science. And on the other end of the spectrum, you have pygmy gobies, which live only about 10 weeks. So a very wide range. And an especially interesting clade is rodents, because it's a very tight group, and it has like a 10x difference between naked mole rats and mice. And these observations are interesting in two ways. One, it shows that evolution can tune lifespan without drastically changing the architecture of the organism, because mice and naked mole rats are about the same size. They have the same organ systems, yet they live very different lifespans. And this shows that lifespan is a biologically tunable feature and not just inexorable wear and tear. And secondly, this allows comparative biology. Comparative biology can leverage these differences to study mechanisms and evolution of aging and lifespan. So maybe you've already noticed, based on the cases I mentioned, that there's a positive correlation between body mass and longevity across species. And actually, the opposite occurs within species, at least for dogs. And yeah, you probably know that larger dogs have a shorter lifespan. And also, I think that might apply to humans, though I didn't find good references for that. But it's been rumored. I mean, one of the main ideas in evolutionary biology of aging is that extrinsic mortality selects against longevity. The rationale being that if you are in an environment where you're likely to be eaten by a lion within the first 10 years of your life, then there's really no point of being able to live to be 100 biologically. And there is some evidence from comparative studies to support that. For example, if you can fly, then you're less likely to be eaten. And it's been shown that bats and birds have longer lifespans relative to their mass, which might be due to the fact that they can evade predation. And also for non-flying species, being nocturnal or foraging in trees or being subterranean also correlates with lifespan, potentially also because of lower predation risk. So ecological context shapes lifespan during evolution. You can also leverage these differences to study mechanisms of longevity. So one study, Eleanor already mentioned it, measured somatic mutations. These are DNA mutations in the intestines of 16 mammal species. And they found that the rate of mutations varies greatly between species, and it has a very strong inverse correlation with longevity. And we also know that that DNA damage, DNA mutations specifically too, are a hallmark of aging, and we know that from mice studies. We know that it's in mice with defective DNA repair mechanisms live shorter. So this is highly suggestive that mutation rates are a strong determinant of species lifespan. And yet another study showing a hallmark of aging, telomere shortening, is correlated with species lifespan. So actually telomere shortening rate. So telomeres are protective caps at the end of the chromosomes, which prevent the DNA from unraveling. So they're physically like stretches of DNA that become shorter with every cell division, kind of like a fuse. And these have been linked, the length of the telomeres have been linked to aging, and this study showed that actually the shortening rate, not the absolute length of the telomeres, but the rate at which they become shorter, is a powerful predictor of organism lifespan. So the faster it shortens, the shorter you live. So telomere shortening is another potential determinant of species lifespan. Of course, this is not an exhaustive list. There are many others. And we've talked about interspecies variance, but there's also variance within a species, of course. In affluent countries, people live on average about 80 years, but some exceed 100, even allegedly 120. And these are called centenarians. And interestingly, there are geographical regions termed blue zones where you have high concentrations of these centenarians. And one of the findings that arose from studying these populations is that there are strong lifestyle factors involved in longevity. These populations are consisting on less processed food. They engage in regular exercise, physical activity, and they have strong family ties and community connections. Also, I think maybe the paper mentioned the sense of purpose. So these are interesting lessons that maybe should apply on a community level rather than individual level. And also there are genetic factors to longevity. So around 20 to 30% of human variance in longevity is actually genetic. And centenarians are enriched for pro-longevity variants in two genes, especially FOXO3 and ApoE. And you may have heard, it was on the press lately, that variants of this ApoE are also associated with Alzheimer's. And lastly, we can also, accelerated aging syndromes can also inform us of aging mechanisms. So these are called collectively progeria. These are genetic defects that cause a phenotype that's aging-like. And two famous syndromes are Hutchinson-Gilford progeria, and Werner syndrome. And the first one is particularly devastating because if you have this disorder, you will likely not survive beyond your teens. And interestingly, both these disorders and some other progeria disorders also involve defective DNA damage and repair mechanisms.
Let's sit down for this one. Hey everybody, my name is Ryan Spangler, and I am currently the Director of Operations at Immuneaging Bio, and also a contributor for the Longevity Working Group at VitaDAO. So today we're going to talk to you about one of our favorite topics, which is immuneaging. Just a quick overview for the talk today. So I'm going to start out, I'm going to lay out just a general overview of the features of immuneaging and the key ways that your immune system changes over time. And then I'm going to expand on what the introduction from Eleanor and go a little bit, still high level, into the different parts of our immune system, but I want to point out a few different cell types that are important to pay attention to when we're looking at how the immune system ages. Additionally, we're going to get into the process of immunosenescence. So this is how your immune system declines as we age. And then at the end, I'm going to quickly go through some ways and talk about how us as a field are thinking about how we can therapeutically target the aging of the immune system and various interventions and current startups out there that are trying to tackle this issue. So the immune system is very interesting because it's very beautifully central to the aging process. If you look at all age-related diseases, whether this is the Four Horsemen, if you've read Peter Attia's recent book, cardiovascular disease, Alzheimer's, neurodegeneration, and metabolic diseases, there's intricately all a immune system component that are either driving or accelerating these diseases with age. And the immune system as a whole is related to many of these diseases that we see with age. So there's two major features that I really want to focus on for this talk. One is immunosenescence. So this is the general term we use for the decline in the immune system as we age. This can lead to multiple different outcomes. Many of the common ones that we know about are weakened antimicrobial immunity. So this is why the elderly get more infections, or we see certain latent viruses popping up again. Think of shingles. Then you also notice with the elderly that they have an impaired vaccine response. Vaccines don't work as well. You might have to dose higher. And you start to see that your immune system, it does not do as good of a job of keeping everything in check and surveying for malignant or non-healthy cells or damaged cells, and this can lead to higher rates of cancer. Also, the immune system plays a very intricate role in how we repair wounds and damage, and because of increased inflammation as we age, our immune system actually impairs the recovery of tissues. The second main theme that we'll get into is a process called inflammation. Along with immunosenescence, what we see is with age, we have this sort of low-level chronic inflammation in some people. And this ultimately leads to an acceleration of all these age-related diseases that I mentioned. So to expand on Eleanor, I just wanted to kind of give a high level of the immune system. And there's two main branches of the immune system. that both contain very diverse cell types with unique functions. So the first is our innate immunity. This is your first line of defense. It has a very fast response, as in hours, but it tends to be less specific. So your innate immunity recognizes essentially all these patterns that have been conserved with evolution. So patterns for damage, patterns for what a bacteria looks like, what a virus looks like, and it can do a few things. You have these cells that are phagocytic cells. Think of them as the Pac-Man of the immune system that can actually engulf invaders and then also signal to the rest of the body to turn up the systemic immune response, like starting fever or recruiting additional cells to a site of infection. Additionally, all the physical barriers are a part of the innate immune system. So think of your gut mucosa, your skin. This is keeping anything you don't want getting into your body. Additionally, cell types I just wanted to point out is you also have these cells that, these include some of the phagocytic cells that can process invaders, find unique antigens, and then kind of hand these off to your adaptive immunity. Your adaptive immunity is kind of, a little bit more high-strung, so it needs things to pretty much be handed to it. So getting into adaptive immunity, this is a lot more specific, but it is slower to respond. It takes days instead of hours, and it recognizes specific antigens. And has the ability to make memory cells so that if you see the same invader in the future, you recognize it, and then you can quickly produce an immune response in the future. So the two main cell types that are very interesting right here are both originate from the stem cells in your bone marrow, but your T cells, they're called T cells because they're matured in immune tissue called your thymus, that's over your heart, which can differentiate into both cytotoxic T cells that can directly kill infected or malignant cells that have a specific antigen, but can also form memory T cells for future immunity. And then your B cells, named B cells because they mature in your bone marrow. These can differentiate into your plasma cells, which are important for secreting antibodies. And you can also have memory B cells as well, so that you're ready for an infection in the future. So I'm going to go a little bit deeper into immunosenescence. So as I mentioned before, this is the process of the immune system decline as we age. And immunosenescence really it affects all parts of the immune system. So how I'm going to look at this is both at the cellular level, looking at how some of these individual cell types get impaired in both the innate and adaptive immunity. But then I'll also get a little bit into the organ level and how specifically the bone marrow changes as well as your thymus gland. So when we look at the immunosenescence of the innate immunity, you start to see dysfunction cell-wide when we look at it. So those phagocytic Pac-Man cells that we talked about, they no longer can engulf bacteria as efficiently. They're also not as good as detecting those conserved markers of pathogens as well as damage. So what this leads to is a much slower or even poor response to infection. Additionally, those antigen-presenting cells that essentially hand off the disease-specific markers to the adaptive immune system, they don't get as, they start to lose their efficiency in actually processing these antigens and handing off. So you can get more of a delay or actually inhibition of from the handoff from your innate to your adaptive for activating that adaptive immune response. Also, your physical barriers can start to degrade and get damaged as you age. So think of, most of you probably heard of leaky gut. So your gut mucosa can start to get damaged, and then you have a lot of antigens that are crossing your gut that you don't want that could cause additional inflammation. Same with your blood-brain barrier of your brain. And what's also interesting is, especially if you start moving towards this more inflammaging-like state, a lot of these cell types can turn on a more inflammatory-like state that could further drive damage accumulation across tissues. So I wanted to take a second to give a shout out to the bone marrow and the hematopoietic stem cells that are deep within the bone marrow. So your hematopoietic stem cells, I'll call them HSCs from now on, they're one of the most important stem cells in your body. So it gives rise to your entire immune system. So you can kind of see this in this very convoluted diagram to the left, but there's really two main pathways these stem cells can go down. One is myeloid, which these tend to differentiate into more of your innate immune cells that are less specific, as well as your blood cells. And then you also have a lymphoid lineage, which makes up a lot of your white cells. This includes the T and the B cells that I mentioned earlier. And what's really interesting here is when we start to age, we start to lose homeostasis. So one of the things that we start to see is the bone marrow starts to be replaced with fat cells. So you can see in the bottom left that you actually start to lose some HSCs and area where you have HSCs in the bone marrow as you get older, which causes impairment to the entire niche. And then you also start to accumulate damage, and as you accumulate damage and inflammation with age, this causes your HSCs to kind of do a wonky thing. No longer do you get an even balance between this myeloid and lymphoid, but you actually start to preferably make more of these less specific myeloid innate immune cells and less of these specific, naive lymphoid cells that allow you to pick up new antigens and respond to new types of infections. So what this ultimately ends up causing you is it's very difficult as you age to respond to new stresses, new pathogens, and build new immunity. So to dive a little bit deeper into both B cells and T cells, we see at the cell level that, as I mentioned in the previous slide, we no longer produce, we have a reduced production of these naive B cells. And we also, as we age, start to have a decrease in the diversity of the B cells we have. So when we're very young, we have, or as we're adults and healthy, we have a very diverse repertoire of B cells that respond to sometimes millions of different antigens. But as we get older, there's certain cells that get outcompeted, you got preferential treatment for certain clones. And what ultimately happens is you have a way less diverse immune system as you get older. And what this really leads to is both the lack of new B cells and dysfunction of old B cells is inability to recognize and respond to new pathogens. But it's also harder to fight off new infections. And because of the proliferation of the cells that are already there, your memory cells, you have an increased risk of both autoimmunity and cancer. This sometimes may present itself like common B-cell cancers like multiple myeloma. So getting a little bit more into the T cells of the immune system. So the T cells are very interesting because they originally originate from the bone marrow. So as we age and we start to get that skew towards myeloid cells, we start to get less naive T cells. But T cells also need an extra step to become mature, and that's in this piece of immune tissue that is right over the heart called the thymus gland. And what is super interesting about the thymus gland is it actually starts to go through something called thymus involution from very early on in life. So it starts to shrink, and a lot of the cells in this organ starts to turn to fat. And the result of this is you're less efficient as you age in maturing and making healthy T cells, and you have a decreased number of new T cells that could respond to unique antigens as you get older. I wanted to quickly go into cellular senescence. So I know this is another topic Eleanor mentioned really quickly, but this is a pro-inflammatory state where cells stop dividing and then they tend to start to secrete these very inflammatory factors. This is senescent-associated secretory, this is normally onset by a variety of stressors, so just the normal wear and tear damage of cells over time, whether that's DNA damage, telomere shortening, or oxidative stress. And this can happen in both immune and non-immune cells, but it ultimately ends up contributing to this low-grade inflammation that we see over age. Which brings me to the topic of inflammation. So we really see multiple contributors for this low-grade chronic inflammation with age. So it can come directly from the dysfunction of our innate immune system, so specifically the phagocytic cells that are already in our tissues. They can pick up a more inflammatory phenotype as we age, and then they just start to go haywire and just secrete lots of inflammation that can cause even tissue degeneration if it goes unchecked. We just mentioned senescent cells, and you can also start to develop inflammatory phenotypes in non-immune cells as well. So an example of this is you start to accumulate certain types of fat that are more pro-inflammatory as you age, and this can really alter tissue, microarchitecture, and affect organs as they age. So moving on from here, a lot of these pro-inflammatory signals, they really will start to drive excessive immune cell infiltration to tissues where you don't want these immune cells, causing additional damage. And it can make other cells move to an inflammatory state as well. So just the summary there is, these defunctional inflammatory immune cells can be very destructive to aging tissues, and it can result in a progressive functional decline in various organs, and it can also lead to all these different age-related diseases that we talked about earlier. So to conclude, I just wanted to get in and think about the question of how we might therapeutically target the aging of the immune system. Being an immune age, this is something we think about a lot, and we're trying to go after right now. So this is not meant to be like a competitive space slide, but just pulling out examples. Here are just 3 examples of approaches that we've seen on the market right now. And what we're currently doing at Immuneage is we're really focused on both the bone marrow and the hematopoietic stem cells. So something that's very interesting, if you take young bone marrow from a young mouse and you transplant this to an old mouse, you see anywhere from 12 to 30% life extension in these animals. So what this suggests is rejuvenating the stem cell bone marrow niche in older animals gives significant health span, lifespan benefits. So what we're doing at Immuneage right now is we've essentially developed a platform to screen for compounds that make the HSCs look younger. And we've already found some compounds that already outperform young bone marrow transplant. Other approaches that seem interesting, so there was a new company recently that was announced, I think just a couple months ago. There's a George Church spinout that is creating a cell therapy to try to rejuvenate the thymus. You know, if you could rejuvenate the thymus, you could restore T cell function and T cell abundance as you age. And then there's a lot of groups that are really just generally trying to target inflammation in general. There's a really cool program at BioAge where they're targeting inflammasome, but there's lots of companies, especially if you look into like neurodegenerative disease space or a lot of these age-related disorders that are just broadly trying to target inflammation. Oh, and that's my last slide, but thanks everyone.